Hey, Mr. P here. This video we're going to talk specifically about enzymes and really everything you need to know about enzymes moving into the higher level content that is metabolism and enzyme inhibition. So let's get started. First thing you need to do before kind of understanding enzymatic action or enzymatic rate of reaction is we have to understand the anatomy to an enzyme and substrate. Okay, so let's kind of get our bearings straight on some of the terminology, some of the language that is kind of needed throughout the video. And so that is enzyme, substrate, and active site. The enzyme is this big structure that is made out of protein. The enzyme is going to act on a substrate, which is typically, in this case, a larger molecule that this enzyme is breaking down into smaller products. It can be smaller products as the substrate for other enzymes that are responsible for building up larger complex macromolecules, but we are using sucrase. Sucrase in this particular example is an enzyme that is used to catalyze the reaction of the breakdown of the substrate sucrose into the two monosaccharide components that are fructose and glucose. Both of those would be individual monosaccharide sugars that are more readily used, more readily accepted by individual cells. And so the enzyme is the large protein complex. This is the particular substrate. The substrate will fit into a particular site on the enzyme called the active site, and there is a certain amount of substrate specificity or enzyme substrate specificity that we will get to in a little bit. But this is an enzyme, this is a substrate, this is the active site. The substrate fits into the active site perfectly in order for the enzyme substrate complex to be made. This particular enzyme is going to catalyze the reaction of converting sucrose into two monosaccharides, glucose and fructose, with the addition of an H2O. H2O will be split into its two components. One H2O molecule will be converted into an OH, and the other one will be an H. Notice H2O is two H's and an O. The OH will bond to one of the eventual monosaccharides. The other H will bond to the other monosaccharide, and so essentially this would have an OH, this would have an H, which you can see here. Here's the H, here's the OH, and you, because of your bonding of these OH and H to the individual monosaccharides, they can no longer hold on to each other, and so they are released as individual monosaccharide products. So the products are released. So 4 steps. Active site is available on the enzyme. The substrate is going to bind to the active site, which makes the enzyme substrate complex. The enzyme actually actively catalyzes the reaction, often with the use of water, and they release the products. And so that is a generic, general enzyme-catalyzed reaction. Now let's get into a little more depth. Enzymes are globular proteins. You can see this structure over here is a 3-dimensional globular protein. These globular proteins are composed of individual chains of amino acids, just like any other globular protein, but these particular globular proteins have a highly developed function or a highly specific function. These globular proteins that are enzymes, or that make up the class of enzymes, or the class of proteins that are enzymes, catalyze reactions. And so just like we saw in that last diagram, here's a different version of the same diagram. We are still catalyzing the reaction of a disaccharide into two monosaccharides. This would be a different enzyme than the enzyme we saw before. In the last slide we saw sucrase was the enzyme that broke down sucrose. Okay, sucrose and maltose are both disaccharides. This particular enzyme would be maltase. Okay, we know it's maltase because it is the enzyme that breaks down the sugar maltose. Hopefully by now you know that the suffix "-ose" means sugar and the suffix "-ase" means enzyme. Often you're going to have a very similar name in both the enzyme and sugar. Okay, maltose is broken down by maltase. This enzyme is going to bring in this particular substrate, bind to this particular active site, catalyze this reaction by breaking the bonds, adding an H and an OH from water to kick out the two products. In this case would be glucose and glucose. Glucose bonded to a glucose makes a disaccharide that is maltose in this case. And so Enzymes are considered biological catalysts. What are catalysts? Catalysts are molecules that do things, initiate reactions. And so if it is a biological catalyst, it is a molecule that is going to initiate biological reactions. The way that it does that is it lowers activation energy and it increases the rate of reaction. I'm going to show you a graph in future slides that's going to kind of highlight this, but for now, know that catalysts lower activation energy and speed up a chemical reaction. And so by these two things, they are actually able to increase the rate or efficiency of a biological reaction. Okay? Increase the rate and efficiency of a biological reaction. How does it do that? It lowers the activation energy, which is the energy necessary to initiate or get the reaction started, and it's going to speed up the rate of reaction once it is started. Enzymes have an active site. We've already established that, and the active site has to be a specific, has a form of specificity. Okay, so specificity. There is a specificity to the active site. The active site has to be specific or have a specific match to a specific substrate in order for the enzyme to, one, know that it is acting on the correct substrate, but also so that it has the right conformation or the right shape to initiate the reaction. Enzymes can be used several times. They can be recycled. They are not a one and done, and we're going to talk about that at more depth in future slides. So, I'm not going to spend time going through this particular diagram. We've kind of already done that, but as a recap, remember you have a substrate. It's going to bind to an active site. That enzyme is going to initiate or catalyze that reaction by adding water, and because it adds water, it splits that H2O into an H and an OH. It's going to give the OH to one, it's going to give the H to the other, and it's going to break the bond between those two monosaccharides to release them as individual monosaccharide products. Okay, globular protein which catalyzes biologic reaction, that is the enzyme. The substrate in this case is maltose. It is a disaccharide. The products that are released from this particular active site are going to be individual monosaccharides in this case, and they have a certain amount of specificity. And so what is a good way to understand this enzyme substrate specificity? And that is through the use of the lock and key hypothesis. What is the lock and key hypothesis? Everybody has an idea of how how locks work. Okay, padlocks in this case require a specific key in order to open or unlock and lock them. And so in this analogy, the key would be analogous to the substrate. It is the actual thing, object, that fits into the key slot. The keyhole in this particular analogy would be the active site. Okay, the keyhole is specific to the shape of the key, just like the active site is the same or is a match to the shape of the substrate. The enzyme is the actual lock itself. And so the lock has a particular site that will accept a particular key, and the particular key is the only key shape that fits not only in the active site but will actively move the lock or release the lock. Are there padlocks made by the same manufacturer in the same line that have a similar shaped key? Yes. There are instances when slightly different shaped keys can fit into the same lock as a different key because they are similar enough, but there is only one key type that will actively engage and disengage the lock. Okay, very similar to kind of the enzyme and substrate complex, or the enzyme substrate specificity. And so we like to use the lock and key hypothesis as a way to demonstrate the enzyme substrate specificity, but also use of an analogy to help understand kind of the importance of matching the shape of a substrate to the shape of the active site. Okay, will eventually unlock the lock, which is what any enzyme is really vying to do, right? It's supposed to produce this enzyme substrate complex based on enzyme substrate specificity, and this substrate initiated the enzyme to do something for it. And so this key was successful in opening this particular lock, much like a substrate fitting into a particular active site on an enzyme, correctly causing the enzyme to do something successful. How do enzymes work really? While to this point I've talked about substrates fitting an active site specifically, okay, to produce enzyme substrate complex, it's not how it technically always works. This induced fit model is a more correct way of understanding the enzyme substrate complex. And what this induced fit model is really saying is that there is a slight deviation between the substrate shape and the enzyme shape. And so the enzyme is actually, while very similarly shaped to the substrate, going to actively change to accommodate a variety of substrates, so that if the substrate isn't an exact match, the enzyme is going to become a match based on an induced fit model. Okay, the substrate is going to be kind of is going to change slightly the enzyme active site shape to accommodate the shape of the substrate. And so the enzyme is going to bond to the substrate. It's going to form the enzyme substrate complex through an induced fit. The enzyme products are going to be produced, and the products will be released, much like that lock and key hypothesis or analogy. But the difference between this and the reason this one is more successful at demonstrating an enzyme substrate specificity is because often this enzyme isn't a correct or an exact fit, much like the lock is to accommodate the key. Okay. Now, a better way of demonstrating this with our lock and key hypothesis is if the key, while being inserted into the keyhole, slightly changed the keyhole or allowed the lock to be induced by or be persuaded by the key in some way. That would be a way of using the lock and key hypothesis as an analogy to this induced fit model. But hopefully you see the difference. Lock and key, it's all about having a particular key shape that is accepted by the lock, and the shape has been determined beforehand, whereas this one, the shape hasn't been fully determined beforehand. I mean, it's a close match, but it's not an exact. And the enzyme is actually going to kind of be persuaded to take on the shape of the substrate as the substrate moves forward. So now that we have a kind of a firm grasp or firm foundation on the anatomy of an enzyme related to kind of like how the enzyme and substrate work together, we need to talk about specifically how the enzyme and substrate come in contact with each other. We know that once they come in contact with each other, they're going to, based on that induced fit model, work based on shape, right, induced fit, but how does the substrate specifically find the enzyme, or how does the enzyme specifically find the substrate? And the way that we're going to answer that or talk about that is kind of this collision theory, right? In a solution, in any solution, the molecules that make up that solution and the molecular motions of the molecules that make up those are constantly in motion. And so within a particular system or within a particular solution, the enzymes that are free in that solution are going to be moving, and the substrates that are free in that solution are going to be moving as well. And so they're going to constantly collide, right? The coming together of a substrate molecule and active site is known as a collision. And these collisions are going to be, in large part, the result of the random movement of both of those individual items within a solution. And so successful collisions or collisions are the ones in which the substrate and active site happen to correctly align themselves, which allows them to bind correctly. And so these substrate molecules can be in any orientation, but in order for this successful collision to happen, this substrate has to perfectly align itself with the active site and then collide with the enzyme in that perfect orientation in order for that that collision to be considered a success. And so if we look at this molecular movement, these substances and substrates within a particular solution are going to constantly be bouncing around. They're going to eventually collide successfully. And we kind of talk about this success as the collision happening and the enzyme and substrate forming that enzyme-substrate complex. And once they form that enzyme-substrate complex, they're going to actually allow the enzyme to catalyze the reaction. And so if this is a particular diagram, much like the diagrams we saw earlier in this presentation, if this is an enzyme and these particular substrates are analogous to a disaccharide like lactose or maltose or sucrose, and this is a lactase or a sucrase or a maltase enzyme, it's going to catalyze the breakdown of these disaccharides into the individual monomer components. And so the way that this particular collision has to act is the substrate has to bind correctly. You'll notice that all of those molecules were in motion constantly. They created a collision. They aligned themselves correctly. The enzyme bonded to the substrate and therefore was able to catalyze the reaction. Once the reaction takes place, the substrates are actually kicked out. Notice the substrates are individual monomers at this point. These monomers came together to produce these dimer substrates, but that collision had happened. This enzyme has catalyzed that reaction, and these products are produced as the result. And so one thing we need to talk about in order to kind of further our discussion of protein and enzyme specificity or enzyme reactionary success is through the idea of denaturation. Okay, protein denaturation is what happens when an enzyme is subjected to particular factors that are kind of outside of the norm, so to speak. And so these globular proteins, and this is essentially a diagram that is showing a variety of tertiary proteins that are coming together to form this globular quaternary protein. This protein has a specific conformation or a specific shape. This protein or enzyme has to be this shape, and this shape allows the enzyme to function correctly. Structure dictates function. I say that all the time, and I'll write it again. Okay? Structure dictates function. Structure dictates function. What does that mean? The structure of a molecule dictates the way that molecule can function. And so if this enzyme is structured this way, it allows it to function a certain way. If this is an enzyme like hemoglobin that is designed to carry oxygen as its substrate in every red blood cell in your body, it needs to be structured this way in order to function correctly. However, if you subject that particular protein to excess heat, let's say you have a raging fever, or you have an increase in your core body temperature due to, let's say, heat stroke or heat exhaustion. Okay, you're outside playing sports, or you're outside working and it's summer and it's really hot. Your body, your core body temperature can actually exceed kind of the normal temperature, optimum temperature of this particular enzyme, and what happens is the bonds that are holding this particular enzyme together and stabilizing the shape or stabilizing the conformation can actually start to weaken and break. And so if these bonds that are designed to produce the specific shape start breaking, then it's not hard to conceive that the shape of that enzyme that is held by those bonds is going to start to change. And when that particular protein changes, the shape of that protein changes, we call that denaturation. Okay, when the bonds that are designed to hold that conformational shape intact, when they start breaking, the conformational shape of the protein changes. It often renders it useless because, again, structure dictates function. If you change the way a particular molecule is structured, you're going to change the way that it functions. Okay? If you change the shape of a shoe, it's probably not going to fit your foot very well. Same can be said with an enzyme. If you change the way an enzyme is shaped, you're going to change the way that enzyme acts with its substrate. And if you change the way it acts with its substrate, it's going to not act with the substrate correctly. Okay? And so as we increase the temp, Let's say we're increasing the temp, we're getting a fever, we're getting heat stroke. Eventually, those hemoglobin proteins are going to change. Notice that these bonds have broken and this protein has kind of become more oblong or more drawn out. This protein is not going to work in the same way that original protein worked. And so that enzyme has been rendered useless. Now, these changes are often not permanent, okay? And so as we start to cool the overall body down or the core body temp down, we kind of come down off of our fever, so to speak. These enzymes can actually revert back to the original conformation and therefore revert back to the original function. Okay, but again, you're going to see it all over the place in a variety of videos that I put out. Structure dictates function is one of the kind of universal themes of biology. You have to understand that. If something is structured a certain way, it is structured that way because it has to function that way. Very simple. Okay, now let's look at specific factors affecting enzyme-catalyzed reactions. We already talked about what happens when optimum environments aren't maintained. Denaturation can happen. Okay, and so these are a variety of factors. We have enzyme concentration, we have temperature, we have pH, and we have substrate concentration. And so we're going to look at all of these in depth. We're going to kind of talk about why the graphs are looked or drawn out or look the way they look. But it's absolutely vital that you understand why these factors exist or why the graphs look the way they do. Okay, so let's start with temperature. Temperature is one of the most important factors that affect enzyme-catalyzed reactions. The reason it is skewed this way is because there obviously is an optimum temperature, and so we kind of break this into two parts. Obviously, there's kind of this ascending, and then there's a descending side of this particular optimum temperature. Why does the rate of reaction increase to the optimum temperature? As we, for instance, increase the temperature to about 37, 36 degrees, we are actually increasing the rate of those molecular movements and therefore increasing the rate of collisions. If we increase the rate of collisions, you're obviously going to increase the chances, naturally, that the substrate and enzyme bond. If you increase those collisions, you're going to increase the efficiency or the rate of that enzyme, okay, or that enzyme biological reaction or catalyzed reaction. Once we exceed this optimum temperature, there's a very, very fine line before the conformational shape of that enzyme or that protein changes. This side, the reason it drops way off very steeply is because as we exceed optimum, this is where that denaturation is going to happen. As we exceed the optimum temperature, there is a maximum temperature before the protein is denatured. And I would say that because this enzymatic rate of reaction is almost at zero, we have denatured our protein. And what is happening from optimum to basically zero enzymatic activity, the conformational change or the conformational shape of that protein is happening very rapidly. Okay, bonds are breaking, the protein's being drawn out. It's being drawn out and it is no longer going to function the way that it's supposed to. So it drops to zero. This is our collisions are increasing to a point where we get an optimum temperature which is maximizing the efficiency of those collisions, leading to optimum. But then after that, it's a very steep slope. We no longer can really keep our enzyme in the proper shape, and so it therefore denatures and it renders our enzymes completely useless, right? So temperature affects the rate of motion. It increases the molecular collisions, and it increases those molecular collisions, leading to increased or higher rates of reactions until optimum temperature is achieved. Past that optimum temperature, the enzyme is denatured, therefore it is no longer useful. But, like I said before, when we lower the temperatures back towards optimum, we can re-establish often protein enzyme function. Okay, really important. Temperature is a very important factor that affects the rate of enzymatic activity. Now if we focus on pH, pH is another one that is greatly impacted or that is greatly impacting enzymatic catalyzed reactions. You'll notice that this graph does look different though. it is not heat or temperature anymore, it is pH. There is always going to be more of a bell curve shape with pH because there is obviously an optimal pH just like there's an optimal temperature. However, because it is a even scale, let's say 1 pH below optimum and 1 pH above optimum often has the same effect of our enzymatic reaction. Okay, so it is obviously causing protein denaturation on both sides of optimum. As we deviate from optimum pH, it is causing those molecular bonds to break, causing the conformational shape of the protein to change, therefore rendering that particular protein useless. And it has to do with the ions that are released in solution. We know that as we decrease pH, we are actually increasing acidity. We are increasing the H+ ions in solution. As we increase our pH, we are actually releasing OH- ions in solution. And because these are charged, they're often going to really or greatly influence the areas of the protein that are oppositely charged, and that causes our protein conformational shape to change. Okay, so when a solution becomes too acidic, large numbers of H+ ions are released. Those often bond with the opposite, right? The negatively charged portions of the active site or substrate, and that's going to cause the active site to change. Again, when we are changing the shape of our protein, that leads to protein denaturation, which leads to an increased or a greater difference for our overall shape, which leads to reduced enzyme-catalyzed reactions. Okay. If we focus on substrate concentration, you'll see that this graph looks substantially different from our other two. These are more bell-shaped. This one obviously almost looks like population growth, where we're actually getting kind of to that carrying capacity, so to speak. But we reach a point of saturation, which is kind of like optimal. But you will see that as we continually increase the concentration of our substrate, once we reach that saturation point, we're no longer going to increase the rate of reaction because we are causing our enzymes to work at their maximum capacity. Just by increasing the substrate that the enzyme is acting on, just by increasing how many substrates our enzymes have to work, isn't going to indefinitely increase the rate of reaction, right? I mean, if we have 10 enzymes, those 10 enzymes are only going to be able to do so much work. You can't just continually add work and add work and add work and just continuously increase the efficiency of our workers, right? There's only so much workload, and there's only a maximum for each of the enzyme. Okay, there's a maximum kind of saturation point for each of the enzymes. Once those enzymes are working at their saturation point, increasing substrate concentration past that point isn't going to do anything. Those enzymes are going to hold firm at their maximum efficiency, right, or their maximum rate. And so we get to optimum or the point of saturation, and then we stay there. If we were to get there and then drop off the amount of substrates, then you would see a reduced saturation point, right? So there's a constant amount of enzymes in that solution. As a result, the concentration of substrate increases, the rate of reaction will increase as well, but it will only increase to a certain point, right? If every enzyme molecule is working as fast as possible, adding more substrates to the solution will not increase the reaction rate any further because they are all working at their maximum rate. Okay, so if we look at the application of enzymes in industry, basically, why do we employ enzymes, or what benefit do the enzymes actually provide us within a variety of industry is really important to understand. I mean, enzymes aren't something that are just abstract, minutia biology. There actually is real-world application of these enzymes to actually better our society and to provide us products that we've kind of become accustomed to purchasing. One of the areas in industry that actually employ enzymes for the benefit of us is in detergents. Okay, detergents that are both clothes detergents or laundry detergents, also dish detergents. All of these types of cleaners are going to contain proteases and lipases. What are these? These are enzymes that break down protein. These are enzymes that break down lipids, which are your fats and oils. Okay, and so all of these are vital to break down when you want to actually clean not only dishes but clothing. Okay, you got to break those things down in order to get them out of the garment or off of the dish that you're trying to clean. Biofuels, okay, getting really important or really popular in today's society. These biofuels are actually produced by enzymes, specifically enzymatic reactions that break down starch and grains into ethanols that we use and can burn for combust for fuel. That is a particular enzyme reaction in order to break those down. Textiles, okay, garments that you buy, the fibers that produce these textiles or produce these garments are often accomplished using enzymes. Blue jeans could not be produced without several different instances in which enzymes are employed. And then within the food industry, there are a variety of enzymes that help us produce the consumables that we've become accustomed to buying. Again, pectin is an enzyme that actually helps increase the juicy yield of fruits in order to produce a more cost-efficient juice drink. Isomerase can actually be used to convert glucose into fructose to be used as a sweetener in a variety of food products. Rennin is an enzyme that is used in cheese production. You cannot produce cheese in a mass production kind of way without employing enzymes like rennin. And then other instances are like paper products. Okay, we are a paper-based society, a paper-based consumer. And so by employing enzymes during the pulping of the wood actually gives us a better quality paper product, but also a higher yield of paper product through the pulping of the wood. So hopefully you can see that there are a variety of uses that are kind of outside of just the area of biology. We actually produce a variety of products that better our society as a whole by employing those enzymes. And so how are those enzymes utilized in an industrial setting that allow us to produce products like what I've already mentioned? And it's through the use of often immobilized enzymes. So we're going to get there. Okay, so immobilized. What does that mean? Enzyme is not dissolved. Often the substrate concentration can be increased, increasing the rate of reaction. We already went through that graph, right? If you increase the substrates being employed, you're going to reach that maximum capacity. We want to we want to work at this maximum efficiency, right? If our enzymes are constantly working at maximum and we can keep this substrate concentration high, we're going to ensure that we're producing our products at a much greater efficiency, which is obviously going to lead to cost savings. When you're a company and you're trying to produce goods, you want to remove as much of your cost as you can so that you can pass those savings on to consumers and you can produce a product at a much more competitive price. Right? We want to increase the rate of reaction. One way we know that we can increase the rate of reaction is by increasing that substrate concentration. We want to allow those enzymes. We want to ensure that those enzymes are working at a maximum efficiency. Enzymes can be recycled. That's really important because if you're a company and you're producing products, you want to be able to employ these enzymes and use them over and over again so that you are producing a cost savings. You don't want to have to continuously produce new batches of enzymes that are kind of a one and done, right? You want to recycle and reuse the enzymes in multiple batches so that you can ensure that you are passing on those savings to your customers, right? Enzymes are more stable to changes in temperature and pH if they are immobilized, okay? Again, that leads to a cost savings because if our enzymes are more stable, then you're going to use less of them, you're going to denature less of them, you're going to go through less of them. That results in a cost savings. Okay, it's really important to decrease the chances of denaturing or degrading our particular enzymes as we work towards producing the products that we've kind of outlined in previous slides. Right? So what does this mean? On top of cost savings, what does it mean? Immobilized enzymes is we are restricting the movement of the enzyme by attaching the enzyme to a specific kind of aggregate material. often aggregations of enzymes are going to be bonded together. We're going to take a lot of enzymes and we're going to bond them together. By bonding the enzymes together or congealing the enzymes in a particular solution, you're going to keep them stable. You're going to lose less of them, and you're going to denature less of them. Okay, that's really important. We can reuse them over and over and over. We are going to often not only bond them together, but we're also going to attach those particular enzyme complexes, or the enzymes that are bonded together, we're going to attach to a surface of either glass, or we're going to attach them into a gel bead. And one of the applications of this of immobilized enzyme specific gel bead application is through lactose intolerance. Okay, we have in the 21st century, right, we've identified that there is a need for lactose-free dairy products because there is an increasing demand. There is an increasing intolerance that is being built to these types of milk sugars. And so what does all this mean? Lactose, as you know, or are probably familiar with, is a milk sugar. Okay, lactose is a milk sugar. Lactose is something that we're not born intolerant to. In fact, we are often, because we're mammals and we rear our young and we produce milk, like other mammals, we are born with the ability to digest lactose. Lactose is the sugar that is found in milk products, and so we have to be able to, one, feed our young milk-based products, but we also have to have the ability to break down that lactose. And one way to break down the lactose, or really the only way innately to break down lactose, is through the production of these natural lactose enzymes called lactase. Lactase is an enzyme that digests the lactose disaccharide into the two monosaccharide components, glucose and galactose. These two monosaccharides are much more readily absorbed into the bloodstream by the individual cells that make up the bloodstream. As we age, because we don't often live on a milk-based diet forever like other mammals, we get more and more susceptible to lactose intolerance. Okay, we cannot readily absorb lactose. We often can't break it down, right? And so when we, if we are lactose intolerant, or we are more susceptible to intolerance of lactose, by consuming lactose, we are actually, in a sense, overfeeding the normal gut bacteria, which leads to the symptoms associated with lactose intolerance, like cramping, excessive gas, diarrhea. All of these intestinal problems are brought about because the normal gut bacteria of the digestive tract are actually kind of overfed. They're producing too much gas product. They're producing too much bacteria-based products, and that leads to kind of a discomfort, and it leads to a deviation from the norm. And if you deviate from the norm, then you're going to experience symptoms that deviate from the norm as well. And so to avoid these unpleasant symptoms of lactose intolerance, we have to have the ability to produce a product that is lactose-free. And so one way of producing lactose-free milk, for instance, is through the process of lactase-impregnated gel beads. And so this is really kind of a simplified version of what's actually happening, but essentially to produce lactose-free milk, we are going to employ these immobilized enzymes. We're going to take these enzymes, we're going to immobilize them. It means we're going to attach them to the gel and attach them to each other. We're going to put them into bead form. We're going to put the beads into a funnel, and then we're going to continuously run the lactose-containing milk, basically the raw milk, normal milk, into the funnel, and the milk that comes out should be lactose-free because the enzymes that are contained within the beads of the funnel are actually going to work like enzymes should, and are actually going to break down the lactose disaccharides into the individual monosaccharides. And so the milk that comes out of the funnel is going to not have lactose, but is going to have glucose and galactose. And because we are producing a milk product that is void of lactose, we're not going to have the same kind of symptoms associated with lactose milk within the gut of a lactose intolerant individual. Okay, so the immobilized enzymes remain to be used again and again. We can use these funnels over and over, and so it does not affect the quality of the milk. It's still milk. It still tastes like milk. It might be a little slightly sweeter just because it has a glucose and a galactose sugar as opposed to the very unsweet lactose. But it does provide a lactose-free product that lactose-intolerant individuals can utilize in order to indulge in the same milk product family of products that everyone else can. And then while we're talking about lactose intolerance, this kind of breeds into evolution a little bit, or natural selection a little bit, which will be later in the syllabus, but it's kind of cool to look at two different maps kind of back to back, or kind of one superimposed over the other, or just a correlation between the two. And on the left, this is a map that shows total milk consumption per capita. Obviously, as you go down this key or this legend, the amount of milk that is going to be consumed is increasing. So we are a very high, based on these colors, a very high consumer of milk. And there's other countries like these Scandinavian countries that even consumes more than we do. And there's a couple places in Europe that ultimately consumes a lot of milk as well. Australia consumes a lot of milk, and then some countries like Asia, specifically China and India, parts of Africa, even parts of South America, don't consume milk at all, or very little milk consumption. When you compare the data or the map to the map on the right, this is actually the incidence of lactose intolerance. So as you go down this legend, the incidence of lactose intolerance is actually increasing, ultimately up to even 100%. And you'll notice that areas where high milk consumption occurs, there is low lactose intolerance. In areas where there is zero milk consumption, there is huge incidence, or very high incidence, of lactose intolerance. I mean, there is a literal inverse between these two, which is very strong. What does that mean? Well, there's two things. One, it's either the population as a whole is very intolerant to milk, and so they don't consume it, makes sense, or they don't consume it because they're intolerant of it. Either way, natural selection's at work. As a population becomes more and more and more and more intolerant, they produce offspring that are more and more and more intolerant, and so the populations are going to get more, as a whole, more intolerant or less intolerant based on the distribution of those genes, distribution of those mutations as a whole population. Pretty cool, but I think that's where we're going to end it. Bring your questions to class. See ya.